Hello everyone, welcome to our next video, and today we're going to discuss about the absolute configuration of amino acids. And in order to know the absolute configuration, we must first know what are enantiomers. So we'll learn about enantiomers and then we will move into the absolute configuration of amino acids that can be actually discussed in two ways. One is the known D and L method, and one is the very modern RS method. So we'll get into both of these. So please stay tuned to watch the absolute configuration of amino acids. So let's get started. We need to know, as I said, what are enantiomers. So in order to know what are enantiomers, we must first know the structure of any chiral carbon atom. So for example, let us take glyceraldehyde as a consideration here. So glyceraldehyde is a chiral carbon center, and this is how it is organized. It has one CHO group, one hydrogen, one hydroxide, and CH2OH. This is one configuration of glyceraldehyde. There is another configuration of glyceraldehyde which we can write like this, with CHO, CH2OH. Here is the hydroxide and here is the hydrogen. So these two have swapped their places, exchanged their places. Now this sort of configuration that we are writing in a 2D plane is called the Fischer's configuration. Okay, so this is how Fischer used to depict molecules. So based on this Fischer configuration, what we can see here is this hydroxide group in this configuration is towards the left. In this configuration, the hydroxide has gone towards the right. So as a result, these two are two different absolute configurations of glyceraldehyde. So how to name them? If the hydroxide group is towards the left, it is called the L isomer, and if it is towards the right, it is called the D isomer. L and D. So this is the L isomer and this is the D isomer, capital L and capital D. Don't confuse these two with levorotatory and dextrorotatory. Those were the optical properties. So that small L and this capital L are not the same. Okay, so this is L isomer. So this will be L glyceraldehyde, and this is the D glyceraldehyde. Now, if you take a mirror and place one mirror out here in between the two, you can see that this image is laterally inverted here. The image of this is laterally inverted in this part. So as a result, this has just formed its image. The D is actually the image of the L, or you can say the vice versa. The L is the image of D. So these are mirror images of each other. But if you try to superimpose one over the other, you cannot. Because if you try to bring it on top of this, you will have the H over the OH and OH over the H. Just like if I try my left hand and I take my right hand and I try to superimpose these will not be superimposed perfectly. Okay, because they are not the same. My thumb will overlap over my little finger. This little finger will come and overlap with my thumb. Only the middle finger will be completely aligned. Otherwise, a ring with the index and ring with the index. So this will not overlap properly. That means these are not superimposable. But if I take a mirror and place one mirror in the middle of my two hands, I can see that this is the reverse of this. So this right hand is the image of the left hand and the left hand is the image of the right hand. Okay, so exactly the same case is happening here. So these are mirror images but non-superimposable. That's why these are called enantiomers. So that's what is an enantiomer. An enantiomer, or actually not one enantiomer, enantiomers always occur in pair or more than two. So enantiomers are those structural isomers that are non-superimposable mirror images. So they will be mirror images of each other, but they cannot be superimposed over one another. These are called enantiomers, and this is one very simple example and one of the most popular examples that people use to demonstrate enantiomers, the glyceraldehyde. Now let us take this into our amino acids and see what happens there. So amino acids also, we have one chiral central carbon, and to this we have four groups attached. So let us take one general structure of amino acid in the Fischer configuration, 2D configuration. So what we saw in the last classes, we have one R group, one H group, one COO minus, and one NH3 plus. So in this configuration, we will consider the N group. So NH3 here is towards the left. So this is an L amino acid. Now if I move the NH3 towards the right, which is also a probable configuration that amino acids can have, this is our D. Since it is towards the right, it is the D amino acid. Very simple, isn't it? We have NH3 towards the left, it is the L isomer. NH3 towards the right, it is D isomer. So left means L and D means right. If you remember only L for left, then obviously D will be for right. So that's an easy way to remember, very easy. So NH3 towards the left, it is L. NH3 towards the right, it is D. One very interesting fact here is that all the amino acids that are formed inside the body, that is all the proteins present containing the amino acids. What I mean to say is whatever the ribosome forms inside our body, means the proteins that are formed from the ribosomes, all these proteins only contain the L amino acids. This is a very interesting fact, and why it contains only L amino acids is not well known, but it is seen that all the protein molecules present inside your body or any animal's body or any plant body, whatever the ribosome forms, it only considers the L amino acids and not the D amino acids. So, but it does not mean that D amino acids don't exist. D amino acids are present. D amino acids are generally found in certain antibiotics. So, antibiotics which contain peptides. Antibiotics are of various forms, so they are peptide antibiotics, and peptide antibiotics contain these D amino acids. Also, another place where you can find these D amino acids is in the cell membrane of eubacteria. In eubacteria, you can find these D amino acids. So this is our configuration of L and D amino acids. Now this was based on Fisher's projection. As I mentioned, there is another type of configuration called the RS configuration, R and S. So let us see what does that mean. So R comes from the word rectus, which is a Latin word, meaning in rectus means it is right, and S comes from sinister, sinister meaning left. So R is for right and S is for left. So how do we use these two? So this is a little bit different from our Fisher. Fisher was very simple. This is also not that tough, but not as simple as the L and D isomerism. So for this, we can't draw the Fisher's projection. Fisher's projection won't work here. So what we are trying to draw here is a 3D conformation. So let's see how to draw that. We'll take the chiral carbon atom here. We have 1, 2, 3 and 4. We have 4 atoms to be bonded. So we have here the NH3, we have the COO minus, we have the hydrogen, and we have our R group. Okay, so what does this mean? This symbol means that the NH3 is coming out of the board. This dotted line means hydrogen is towards the inside of the board, R group is towards the top, and COO minus is towards the left hand side. So these two, R and COO, are in this plane. One is towards the top, one is towards the left. NH3 is coming out of the board and H is going inside the board. Okay, it is towards the back. Hydrogen is towards the back. So hydrogen is, if I take these 4 and I try to show you how it is formed. So it is like this. So this is coming out of the board. This is my NH3 coming out of the board. This is my hydrogen which is going towards the back of the board. This is my R group on the top, and this is my C which is on the same plane as the R group. So this is forming a tetrahedral structure. So now let me try which one was my marker. Okay. So sorry. Now if we try to go back into this, what we are trying to do here is we are trying to give a priority number. So what is a priority number? How do we give a priority number? So in order to understand this, let us remove the R. Let us consider any particular amino acid. A very simple amino acid is the alanine. So in alanine, the R group is actually CH3. CH3 is the R group in alanine. So Let me erase this one, it will be easier for us to understand. This CH3 group is different from the others, so we still have a chiral carbon center. This is just for now, we will get into the different types of amino acids, so don't worry about that. I am just taking one example of alanine, where alanine, the R group is the CH3 group. Okay, so in this, now we have to give priority numbers. So priority numbers will be based on the atomic number of the atoms that are attached to the chiral center, chiral carbon center. So here we have 4 groups, right? In the first group we have carbon, which is CH3, where carbon is attached to the carbon directly. Here nitrogen is attached directly to the carbon, here hydrogen is attached, and here also carbon is attached. So we will see which elements have what atomic number. So higher the atomic number, more priority; lower the atomic number, less priority. So here let us consider CH3. In CH3 the carbon or atom directly attached to this carbon is carbon. Here carbon is attached, so carbon's atomic number is 6. In case of NH3, nitrogen is directly attached to the carbon, so nitrogen has atomic number 7. Hydrogen is directly attached to carbon, so hydrogen has atomic number 1. And then we have COO-, which also we have carbon which is directly attached to this carbon, so this carbon atomic number is 6. So highest atomic number goes to nitrogen, so nitrogen gets the first priority, so nitrogen will be our number 1. Okay, now which one will be second? This is also 6, this is also 6. So what to do with this? So if your first atom cannot help you give the priority, we move to the second atom. So in case of COO-, second atom is oxygen, so oxygen's atomic number is 8. In case of this, the second atom is hydrogen, hydrogen has atomic number 1. So since oxygen has higher atomic number, so this one will get our second preference. Okay, so this one becomes number 2, and automatically this one becomes number 3, and hydrogen having the lowest atomic number becomes number 4. As of now, I hope it is clear. We have just given the priority numbers based on the higher atomic number. Okay, so here now we will try to move from number 1 to number 2 to number 3. So let's see how we do this. We are trying to move from 1 to 3. So if my direction becomes clockwise, then it will be called the R configuration. If it becomes anti-clockwise, it will be called the S configuration. So let's see what happens here. We are here at number 1. We want to go to number 3, but we have to go through 2. 2 must be in the middle. So in order to reach 3, I must go towards this side. So it will be like this, where this is number 1, number 2, number 3. I have moved in this direction. So this is a clockwise direction. I hope it is clear. This is a clockwise direction, and clockwise direction means the configuration is R alanine. So this is alanine and its configuration is R alanine. Okay, I hope this is very simple. Just a little one step too much we have to do compared to the DL and isomer. There only we have to do the structure, left and right we understand, but here we are not doing this. We are taking into consideration this whole thing. We are first giving priority to each and every compound attached to the chiral carbon center, and then we are trying to move from number 1 to number 3. Number 4 is not in consideration. Okay, don't care about number 4. Let number 4 stay in its place. We will move from 1 to 3, and in all the amino acids except for glycine, which we will see later, always this hydrogen will be number 4, because there is no other element which will have a lower atomic number compared to hydrogen. So hydrogen will always be number 4, and hydrogen will always stay in this position. So you don't have to worry about this. We have to consider only these 3, and based on 1, 2, 3 we will move, either it will be towards the right or it will be towards the left. So that will help us to understand how these organizations help in getting different configurations, R or S. Now looking at this, if I try to say whether it is D or L, what do I get? Since NH3 is towards the right, CW is towards the left. Okay, CW is not a matter of concern. NH3 is towards the right, so right means D. So this is D alanine as well. So in both configurations we can write this. In terms of DL, this is a D alanine. In case of RS, this is R alanine. Now all the L amino acids, L configurations, all the L amino acids, we have the S configuration. For all the L amino acids, we have the S configuration, and for all the D amino acids, we have the R configuration, except for cysteine. Cysteine is different. For cysteine, the L cysteine has R configuration, which is different, because for every other L amino acid we will have the corresponding S amino acid. So L and S go hand in hand. Okay, L amino acids and S amino acids will be having, means whatever amino acid containing the L configuration will also have the S configuration, and whatever has the D configuration will have the R configuration, except for cysteine. In cysteine, the L cysteine has the D cysteine configuration. This is the only difference between all the others. Okay, so this is for some extra knowledge. So if suddenly you get a question where you have to determine, you are given a question based on this L cysteine, what will be the corresponding configuration in D or R. So remember that L is generally, L corresponds to S and D corresponds to R, but in case of cysteine, L corresponds to I'm sorry, I gave a wrong one here. L will correspond to R. Okay, L will correspond to R. I am extremely sorry for this mistake. L means L will go for R for cysteine only. Okay, cysteine will have L and R corresponding to each other. Otherwise, L always goes with S and D always goes with R. Remember when I first started the lecture, I said L is for left, and during S and R also I said S means sinister, which means left. R goes for right and D also goes for right. So which is pretty understandable. L and S go hand in hand, R and D go hand in hand. But in case of cysteine it is different. L and R means L cysteine and R cysteine go hand in hand. So cysteine is an exception. So that is all about our configuration chapter or configuration lecture. I hope it was clear. If you have any confusion regarding this, please comment down in the comment section. I will be happy to help you, more than happy to help you, because I want things to be clear for you so that you don't have any confusion. So any nitty-gritty things, if you have any confusion, you can ask directly in the comment section. I will be there to answer to the questions. If required, I will make a video on that. If I think the confusion is too big, then I will make a clarification video on that. So I hope everything here is clear. Very simple stuff. Hope you understand. See you in the next one. Peace.